Hello, good morning. Hope you're all doing well by God's grace. So, this morning we are going to have a look at another article or another paper titled A Comparative Study of Different Laboratory Storage Conditions for Enhanced DNA Analysis of Crime Scene Soil Blood Mix Sample. So, this article compared different laboratory storage conditions for crime scene soil blood mix sample. So blood sample that has been mixed with soil from the crime scene. So background. The world of crime is a complex place. A crime can be committed everywhere, such as workplaces, schools, places of residence, vehicles on the streets, on water bodies, and even on the internet in modern times. Every forensic process starts from the crime scene in order to obtain evidence for analysis. Then the aim of an investigation that follows the commission of a crime is to interpret correctly the facts, reconstruct the events, and know what happened. So, blood provides a good ground for the growth of microbes, and these microbes secrete biochemicals which degrade or destroy DNA in the blood. At outdoor violent-related crime scenes, many situations may cause human biological evidence from the suspect or victim to be deposited in the soil. This evidence can be analyzed using DNA to link the suspect or victim to the crime scene. Microbial activities in soil can degrade blood DNA after deposition, making the DNA unusable for profiling. When tissue samples are exposed to harsh environmental conditions, DNA degradation occurs rapidly to the extent that DNA becomes unrecoverable. Various problems can occur from the analysis of degraded DNA samples, and these include signal loss, peak imbalance, and allele dropout. Hence, crime scenes need to be processed as fast as possible and tissue samples properly stored. Quantifier trail kit. So this is the kit that the scientists or the researchers use for their DNA quantification, the quantifier trail kit. The quantifier trail kit quantifies human DNA and total human meal at the same time. So it has three targets: the male target, the large autosomal, and the small autosomal targets. Results from real-time PCR with quantifier tool can assist the analyst to know. So the analyst, by using quantifier toolkit, you can know this stuff. One, if there is sufficient human DNA and or human male DNA for subsequent STR analysis. You can also know the quantity of sample to use for PCR amplification. Then you can also know the ratio of male to female in mixed samples, especially sexual assault samples. Then you can also know the quality of DNA in terms of degradation and the presence of PCR inhibitors. So let's go to the problem statement and justification. That's the problem that the scientists tried to solve and why they did this research in the first place. Bloodstains are often encountered on weapons, clothing, and other materials, and sometimes on the body of the victim. When these materials are discarded at outdoor locations, they may come into contact with soil. Biological sample mixed with soil is often contaminated with material that pose a threat to the DNA profiling process. It is estimated that 10 billion microbes can be found in a gram of soil, and they comprise thousands of different species. These soil microbes destroy DNA in biological samples in a short period of time by fragmenting and poses a threat for successful DNA profiling. Now, crime scenes containing biological evidence are usually processed early to prevent loss or destruction of evidence. Microbes in the soil, organic matter, and storage temperature can all affect the evidence. Evidence submitted to the laboratory from crime scenes are usually not processed immediately due to the large number of pending cases, unavailability of analysts, or unavailability of reagents. There is thus the need to investigate the proper way of storing this evidence at the laboratory to prevent destruction prior to their processing. And the objective of this study was to do a comparative evaluation or a comparative study of three laboratory storage conditions on soil blood mix samples found at crime scenes using DNA analysis. Materials and methods. So let's look at how they did the work. Blood sample. Fresh adult male whole blood sample from a single person in a tube with EDTA anticoagulant was obtained from Dublin Hospital, an accredited private hospital in Nakenoda in the eastern region of Ghana, and transported on ice to the lab. Identity of donor is not known, and sample collection was done by a staff of the facility. So, soil sample. Dry black soil was collected in paper envelope from a garden at the compound of the Ghana Police Forensic Lab and brought to the lab. Storage facilities. Room with an air condition of 25 degrees Celsius temperature, fridge with temperature set to 4 degrees Celsius, and a freezer with minus 20 degrees Celsius temperature serve as the three sample storage conditions for this study. Then reagents and instruments for the DNA profiling. Profiler forensic DNA was used for the DNA extractions. Quantifier Trio DNA quantification kit and 7500 real-time PCR machine manufactured by Blackberry Systems were used for DNA quantification. Then, with the DNA electrophoresis, global filer PCR amplification kit was used. Study time. The study was conducted at the Jubilee Hospital and the Ghana Police Forensic over a period of 12 weeks. So, sample preparation and storage, sample preparation prior to DNA analysis, and sample identification numbers. So, let's look at the DNA quality assessment. So, after extraction, how they assess DNA quality. So DNA quality in terms of degradation was assessed by applied biosystems quantified through kit using the 7500 real-time PCR and following the manufacturer's protocol. Degradation was assessed by comparing DNA concentration and degradation index of a sample from a particular storage time to the previous storage time. Results. So, assessing DNA degradation at the three storage conditions. This assessment was done to determine whether the three storage conditions were able to preserve the integrity of samples in terms of their DNA concentration and quality as storage time progressed. DNA concentrations and degradation indices were compared among the various storage times, and it was observed that DNA concentrations and quality decreased with increase in storage time from room temperature and 4 degrees Celsius stored samples. Minus 20 degrees Celsius stored samples had increase and decrease in concentration for samples with increase in storage time, but DNA quality remained intact. So these were the three laboratory storage conditions: room temperature, 4 degrees Celsius, and then minus 20 degrees Celsius. So assessing DNA degradation at room temperature storage. So samples 2pl, which one is sample 2pl? Sample 2pl, this is sample 2pl. That is the 2-week sample stored at room temperature. So sample 2pl high degradation index is less than 1 at 2 weeks storage. Its concentration dropped compared to the control sample. So they initially extracted DNA to serve as control before they started the whole experiment. At 4 weeks of storage at room temperature, sample 4pl recorded degradation indices less than 1, indicating DNA quality was still intact. Its concentration dropped compared to previous sample. So the quality as time progressed at room temperature storage, the quality decreased with time, but then the quantity was still intact or concentration was still intact. At 6 weeks of sample storage, sample 6pl showed amplification for all targets. Its degradation index was between 1 to 10, an indication that DNA was slightly to moderately degraded. Its concentration was lower than 4-week store sample. So at this time, the concentration also started to decrease. All samples had degradation indices between 1 to 10 at weeks 8 to 12, an indication that DNA was slightly to moderately degraded. Their DNA concentration kept on reducing week after week. There was significant difference in DNA concentrations of samples stored at room temperature compared to the control samples. Then, assessing DNA degradation at 4 degrees storage. At 2 weeks of sample storage at 4 degrees Celsius, DNA concentration of sample 2P4 reduced drastically compared to the control sample. Its degradation index was between 1 to 10, meaning DNA was slightly to moderately degraded. So for the 4 degrees storage, just after 2 weeks, the degradation index was between 1 to 10, meaning DNA was slightly to moderately degraded. At 4 weeks of storage, sample 4PL had its degradation index between 1 to 10, and its concentration was lower than the 2 weeks extracted sample. At 6 weeks, the sample recorded degradation index between 1 to 10, and its concentration was lower than the 4 weeks extraction or 4 weeks storage. At 8 weeks of storage at 4 degrees Celsius, the sample recorded degradation index between 1 to 10. Its concentration dropped compared to the 6 weeks extraction. At 10 and 12 weeks of storage at 4 degrees Celsius, all samples showed amplifications for the internal positive controls only. Reduction in DNA concentrations of 4 degrees Celsius stored samples were very significant compared to their control samples, with p-value of 0.000. Assessing DNA degradation at minus 20 degrees Celsius. So, assessing the concentrations of the samples, there was no particular pattern in terms of decrease or increase as storage time progressed. Degradation indices of these samples were less than 1, meaning DNA quality was still intact even at 12-week storage. There was no significant difference in DNA concentrations of samples stored at minus 20 degrees Celsius compared to the concentrations of their controls. P-value 0.324. So now comparing DNA degradation among the 3 storage conditions, that among the room temperature storage, 4 degrees Celsius storage, and minus 20 degrees Celsius storage. Samples stored at room temperature decrease in concentration as time of storage increased, with concentration decrease being significant. At 6 to 12 weeks of storage, degradation indices of the samples rose to 1 or above. The samples stored at minus 20 degrees Celsius did not follow a particular pattern in terms of DNA concentration as time of storage increased, with no significant difference in concentration compared to the control samples. Their degradation indices were less than 1. Soil blood mix samples stored at 4 degrees Celsius decreased in concentration drastically, and at 2 weeks of storage, their degradation indices were between 1 to 10. At 12 weeks, there were no amplification for targets except IPC, meaning the DNA had degraded drastically. Significant difference was seen between the DNA concentrations of samples from room temperature and 4 degrees Celsius storage. Again, significant difference was seen between the DNA concentrations of samples from room temperature and minus 20 degrees Celsius storage. There was significant difference between the DNA concentrations of samples from 4 degrees Celsius and minus 20 degrees Celsius storage. So DNA concentrations in nanogram per microliter and degradation indices from soil blood mixed samples are the 3 storage conditions. So these are the concentrations, degradation indices, concentrations for samples stored at 4 degrees Celsius and their degradation index, likewise for minus 20 degrees Celsius samples. And you can see from the graph here, DNA concentrations of room temperature stored soil blood samples for the 12-week period. So this is the control. So the concentration kept on decreasing, decreasing. But then the quality was intact in terms of degradation. So the concentrations were decreasing till the 12 weeks. Then for the 4 degrees stored samples, this is the control. At 2 weeks, concentrations had decreased drastically. By 4 weeks, concentration kept on decreasing. Then by the 10 to 12 weeks, there was nothing in there at all. DNA had degraded. Then minus 20 stored samples. You can see there was no pattern here. So it means the DNA was still intact, even at 12-week storage. So this is also the comparison. DNA concentrations at the 3 storage conditions over a 12-week period. So the blue is the room temperature, 4 degrees Celsius, and then minus 20 degrees Celsius. So you can see the comparison here with the minus 20 degrees Celsius always having the higher amount or concentration of DNA, followed by the room temperature. And then 4 degrees Celsius performing very poorly. Detected alleles, that's after electrophoresis, for samples stored at the 3 conditions. Extraction for samples stored at room temperature and minus 20 degrees Celsius all give full STR profiles. At 12 weeks of sample storage at 4 degrees Celsius, there were allele dropouts. At 6 weeks of storage onwards, that's at 4 degrees Celsius, there were no alleles observed for the extracted sample stored at 4 degrees Celsius. So table 3. So this is the table 3. Number of amplified and detected alleles for soil blood mist samples stored at the 3 storage conditions. So for room temperature, minus 20, full profiles, even at 12 weeks. At 4 degrees Celsius, full profile at 2 weeks, allele dropouts, so resulting in 42 alleles at 4 weeks. Then from 6 weeks, no alleles at all. DNA had degraded. So discussion. DNA extraction for clean blood and soil blood mixed sample controls as amplification for all types. This indicates that DNA was not degraded. So let's go straight to, so this is the profile. There's a program of positive control. There's a positive control, and this is the negative control, meaning there was no contamination. So what conclusions did they draw from their research? They came to a conclusion that storage time had effect on DNA quantity and quality of samples stored at room temperature and 4 degrees Celsius. Hence, storing soil blood mixed samples at minus 20 degrees Celsius was the best in this study, while storage at 4 degrees Celsius was the worst. So if you have this kind of sample, soil blood mixed sample from the crime scene, it's always best at the laboratory to store them at minus 20 degrees Celsius compared to room temperature storage and then 4 degrees Celsius storage. So now let's look at the summary or the abstract. Crime scene investigation is an important step in the entire criminal investigation process because this is where evidence is gathered. Blood from the perpetrator or victim of a crime can be left at crime scenes or transferred to other materials such as clothing, knives, and guns. Most often, this body fluid is contaminated with soil at outdoor crime scene, but this might be the only or the most important evidence in solving a crime. This work aimed at identifying the most appropriate method of storing crime scene soil blood samples prior to analysis at the laboratory. So human blood was mixed with soil and stored at 3 different storage conditions, that is room temperature, 4 degrees Celsius, and minus 20 degrees Celsius. Samples stored at room temperature saw significant reduction in DNA concentration as storage time increased. Samples stored at 4 degrees Celsius saw a drastic decrease in DNA concentration just after 2 weeks of storage. By the 8th week of storage at 4 degrees Celsius, there was no detectable DNA. Samples stored at minus 20 degrees Celsius recorded no specific pattern in decrease or increase in DNA concentration for the entire 12-week period. There were full STR profiles generated for room temperature stored samples and minus 20 degrees Celsius stored samples throughout the study. There were full, partial, and null profiles generated for 4 degrees Celsius stored samples, depending on the sample storage duration. In conclusion, -20 degrees Celsius was identified as the best storage condition for soil blood-based samples, followed by room temperature and 4 degrees Celsius respectively. So thank you for your audience, and don't forget to subscribe, hit the button so that you'll be notified anytime I upload a new video. Thank you and enjoy the rest of the day.